Wow, a great start. And our next is Abem, and and Abe is a rising sophomore at Howard, working in the Ferreira laboratory, another neuro-oncology laboratory. You ready? Okay, come on up.
Hello, everybody. My name is Abem Mare. Thank you so much, Dr. Ellen Bogan, for your introduction. I was very lucky and blessed to have had the opportunity to intern in Dr. Ferreira's lab this summer, where I got to do a whole exome sequencing analysis of skull-based chordomas, which then later revealed possible JAK2 related mechanisms. So a little background on chordomas. They're rare bone tumors that arise from the notochord remnants, and they're usually found at the skull base and the sacrum. They're typically slow growing, and they can become very aggressive and difficult to remove. They're also very resistant to chemotherapy. They have a high recurrence. They can become very large, press on the brain and the spine, which later has people get nerve damage. And currently, there are no FDA-approved drug treatments for chordomas. So surgery is the primary treatment, and they're very extremely technical procedures because of the locations being in the base of the spine and the skull, which also later contributes to having a higher recurrence rate after removal. And another form of treatment is radiation coupled up with surgery. Since there are no targeted treatments for chordomas as of this moment, by interpreting this whole exome sequencing data that we can identify any possible driver mutations and dysregulated pathways in these chordomas to improve patient outcomes in the future. So whole exome sequencing was performed on 45 patients' chordoma samples, which then was analyzed using bioinformatic tools like R and whole open source tools. And out of the entire 45 patient cohort, 7 out of the 45, which was 16%, had the JAK2 mutation, while the other 38 didn't have it. And although 16% might seem small, it's actually a great representation because JAK2 is an oncogene, which means it's basically a mutated gene that has the potential to cause cancer. After I used R to differentiate JAK2 into a wild type and mutant cohort, then I performed a gene ontology and pathway enrichment analysis for both of these cohorts. So JAK2 is a ligand in the Notch signaling pathway, and it plays a huge role in cell proliferation and differentiation. And the graph on the top right here is a Kaplan-Meier curve, which shows the survival probability of patients with the JAK2 mutation, and it's between the mutant and the wild type cohort. And we can see that the mutant cohort has a very high probability, and the graph on the bottom right is a lollipop plot, and 3/4 of the mutations land on the EGF calcium domain. And some studies suggest that the distinct inter and intercellular calcium signaling pathways may be mechanisms by which EGF modulates the growth and the migration of these tumor cells. So to assess the functional impact of mutation in our cohort, I performed a gene ontology, enrichment analysis, and I classified these into the biological processes, the cellular components, and the molecular functions. And notably, the mutant cohort showed a lot of enrichment in the cilia-related processes, suggesting potential differences in pathways like the Hedgehog signaling pathway and the Wnt signaling pathway, and that might influence the tumor biology and overall treatment responses. Here I performed a pathway enrichment analysis, which basically means that I used Reactome and Wiki pathways, and I took the most enriched pathways from these and did an oncoplot. On the Reactome side, we can see that the most enriched pathway is the zygotic genome activation. And you can see 60% of the samples harbor the CRUB-BP and the YAP1 mutation. YAP1 is a core effector of the Hippo signaling pathway, which is involved in the development, growth, and repair in general. And the gene is also known to play a role in the progression of multiple different cancers, and it can also function as a potential target for cancer treatments. And then on the wiki side, we see that the hedgehog signaling is the most enriched pathway, while 60% of the samples harbor the CREB-BP and the JMJ-D1C. And this fits perfectly with our earlier gene ontology results, showing the cilia-related processes, since hedgehog signaling pathway is cilia-dependent. For the mutant cohort, the notch signaling pathway is the most altered pathway in the data set, while the hippo pathway is present in 83% of the cases, and the Wnt pathway is not a lot, with 16% of the case, around 17% of the cases. And these pathways are significant because notch, JAK2, which is a notch ligand, can harbor activating mutations that enhance the notch signaling, which can promote the tumor growth in general. And the dysregulation of the hippo pathway can increase the YAP1 activity, which can cooperate with JAK2 to drive oncogenesis. And the Wnt pathway, which is critical for the notochord development, when dysregulated in chordomas, may represent a reactivation of developmental processes that can fuel the tumor growth later on. And when we look at it in the therapeutic perspective, the drugs like isoflavone, curcumin, and withaferin A have been seen to target the notch signaling pathway, and drugs like vetoporfin target the YAP signaling, the YAP gene in the hippo signaling pathway, while the dobutamine targets the hippo signaling pathway in general. And drugs like vantictumab, OMP54F28, have been currently are actually in clinical trials because they're seen to target the Wnt signaling pathway. This is my thank you slide. Thank you so much, everybody, for taking your time to listen to my talk. Thank you to my family for supporting me, to my cohorts as well. And most of all, thank you to Dr. Ellen Bogan, having given me this opportunity as a first-gen student to be able to do all of these things that I probably wouldn't have done if it wasn't for this program. And thank you to all my mentors and people in the lab to